Hi, I'm James Reimer from the University of the Ryukus in Okinawa, Japan, and I'm going to talk about recent discoveries of new taxa of colonial anemones from museum collections. Now, this might not sound like an exciting topic, but I'm going to try to make this as exciting and as interesting as possible. And so on the front cover, you can see a picture of these colonial anemones. So let's learn more about them. And I'd just like to say, this talk and this research would not have been possible without the collaboration of all of the people's names and institutions you see below. Okay, so colonial anemones. First of all, what are they? They're related to corals and to sea anemones, and they're found in a lot of different marine ecosystems. A lot of different species are actually epizoic. That means they live on top of other marine animals. And for this reason, they're quite interesting. However, they're generally not well studied. This picture here shows you one colonial anemone. It looks like a sea anemone, but it's a colony, hence the name colonial anemone. I'm going to call them zooantheria in this talk. You can see two rows of tentacles, you can see the mouth, and then you see the sandy bit that's connecting these different polyps or these different anemones together. So it's actually one animal with multiple polyps or multiple mouths. And this picture here in particular shows a species of colonial anemone that's growing on top of an antipatharian or black coral. Colonial anemones are found on a lot of different animals, as I said before, and this includes antipatharians, black corals, hydroids. You find them on octocorals, or these are gorgonians, closely related to soft corals. You also can find them on worm tubes, and you can find them on the substrate in coral reefs. So these animals are all over the place, and they're often hitchhiking on other different groups of animals. If you go to a museum, for example, these are pictures of Naturalis Biodiversity Center in the Netherlands. You see rows and rows of specimens collected in the past from different expeditions or by different scientists. This might not look too interesting to you. It just looks like rows of jars or something. But if you zoom in a little bit, you can see there's lots of different specimens. And if you actually zoom in on each specimen, you can see there's a scientific name, there's the date, the location that it was collected. And so these collections often house thousands and thousands of specimens, dating back tens to hundreds of years ago. So to a geek, marine biodiversity geek like myself, this is super exciting. For zooantheria, most museums have zooantheria specimens. A lot of these specimens, because not many people are studying them, are understudied. And the other thing that's really interesting, as I said before, is because they're hitchhikers, a lot of specimens of other taxa actually have zooantheria on top of them. For example, a fellow of mine, a research fellow of mine, went to this collection in Naturalis, and they examined over 10,000 sponges. And they found 200 or 2% of the specimens actually had zooantherians. So when we go to a museum, not only do we have to identify the zooantherians, but we actually have to look for zooantherians in other groups of animals as well. So it's a lot of work. That said, these museum collections are huge resources, and I'm going to introduce to you very quickly today two papers where we have found some interesting things from museum specimens. The first one is a paper that we wrote last year, and we were looking at deep sea specimens that are associated with hermit crabs. These things actually form a colony that's shaped like a shell for the hermit crabs to live in the deep sea. And we actually described 3 new species from Australia and Japan. And one of the fun things about working with biodiversity and discovering new or undescribed species is you get to give these species names. We named this one Epizoanthus xenomorphoides. Then we had Epizoanthus australis, and then Epizoanthus gorgonus. Now, you think about these names, australis means south, and it was found in Australia. You know, this makes sense. This is a good name, I think. And then gorgonus, this is from the Medusa or the Gorgon from Greek mythology, because we thought that it kind of resembled snakes around this sort of central area. But the one name I think that we had a lot of fun with was xenomorphoides. If you happen to be a science fiction fan, you'll probably know the xenomorph facehugger from the movie Alien, 1979 science fiction movie. And we named the species after the xenomorph in the picture there. So you get to have a little bit of fun with your science too. The other thing I'm going to quickly talk about today is some work we did in the Dutch Caribbean, which is beautiful, fantastic place to go. Hopefully after Corona becomes less of a problem, we can go there again. And again, we looked at some deep sea specimens as well as some shallow water specimens. And these specimens dated back almost 50 years. We found one species that was in shallower water that we described this year as Parazoanthus atlanticus, but we've also found 3 more species that still do not have names that we're working on now. One, the one on the right, Umimyanthus, is from shallower water, but the other 2 are from deeper water. And so these museum specimens, even if they're collected a long time ago, really harbor a lot of undiscovered or undescribed biodiversity. And so this, to me, makes these collections huge importance and very much treasures. So in conclusion, what can we say? Well, museum collections are obviously valuable resources, especially for groups of animals that are not being well studied now, because we can still collect these animals, and in the future, maybe someone else who has more knowledge can come and look at these animals and describe them or understand their diversity more. And without an understanding of diversity, it's really hard to protect or conserve animals too. Just to give you an overview of what we've done, until 2007 for the groups of Zoanthera, we had, based on DNA from specimens in museums and specimens that we collected in the field, we knew of 4 families and 10 genera. But thanks to all of this work with museums and specimens and all of our collaborators, now we have a much better understanding. We're at 10 families and 30 genera, with more coming. So it's clear a lot of these groups of animals are much more diverse than we thought. And a lot of this is thanks to networks of museums and collaborators. And the last thing I'd like to say, it's clear, looking at all the different people that helped us on this research here, you cannot do biodiversity research alone, and collaboration and working together is very, very important. And so with that, I'd like to say thank you very much and enjoy International Humboldt Day. Thank you very much.